So next biomolecule is nucleic acids, and nucleic acids are the biomolecules responsible for the different control and coordination of metabolic and genetic functions in organisms. So all the organisms, whether animals or plants, all the metabolic functions like protein synthesis, genetic function like transmission of hereditary characters, they all are controlled by the nucleic acids. So nucleic acids basically they provide the footprint of an organism right from the conception. Now nucleic acids are of two types. There are two types. First is DNA and second is RNA. So DNA deoxy ribonucleic acids, and this is ribonucleic acid. Now coming to the real statement on the definition of nucleic acids. Nucleic acids are polynucleotides. This is simple. They are polynucleotides. Polynucleotides. Now we'll see the basic components. They are made up of three basic components. Three basic components are used or involved in the nucleic acids, and they are pentose sugar. Like here we have in DNA deoxyribose sugar, here we have ribo or ribose sugar. So pentose sugar means they have 5 carbon. They have 5 carbon atom. They have 5 carbon atom. Then coming to phosphate group, they have the phosphate group. And third is the nitrogen bases. So these are very much important part. They are the component of the nucleic acids, whether it is DNA or it is RNA. Now nitrogen bases, they are of two categories. Nitrogen bases have two categories. One category is of purines. Purines is a group of compounds. Purine, the group of organic compounds of nitrogen, purines. And second is pyrimidines. Purines are of two types. Purines, adenine A, and second is guanine G. Adenine is 1, guanine. So this is 1, this is 2. Now pyrimidines, they are of three types. Then pyrimidine first is cytosine C, then thymine T. These are the symbols to represent, and third is the uracil U. So these are the symbols how we represent. So the nitrogen bases, purines and the pyrimidines, so they are the categories of the nitrogen bases. Purines, they are of two types. Adenine, and the symbol is capital A. This guanine symbol is capital B. Pyrimidines is other class of organic compounds, and this cytosine C, its symbol is, then thymine capital T, uracil capital A. Now, in DNA and RNA, only four nitrogen bases. Only 4 nitrogen bases are present. Only 4 out of these 5. Out of these 5, only 4 nitrogen bases are present. So, DNA has adenine, thymine, cytosine and guanine. This is in DNA. Adenine, thymine, guanine and cytosine. RNA, it has what? RNA have uracil in place of thymine. So A, adenine, uracil, guanine and cytosine. So in RNA, thymine is replaced by uracil. So this is the basic component, basic composition, how the nucleic acids are made. Now, this was the basic composition of the DNA and RNA. Actual point lies with the formation of nucleotides and nucleosides. Nucleotides, and there is formation of nucleosides. So, as you know, the nucleic acids are polynucleotides. They are polynucleotides. So first, I am going to draw some very important part. And before this, there is one point that nucleic acid molecules are made up of strands. The strands are the sequences. The strands are the sequences. The strands are the sequences. The particular order, sequences or particular order in which nitrogen bases, sugar means pentose sugar, and phosphate groups are attached. This is the meaning of strand. Now, DNA is made up of two strands. DNA made up of two strands. RNA made up of one strand. A particular pattern, particular chain. Now see how the nucleotide is formed. Nucleotide is basically a strand or a sequence. So here S stands for sugar, pentose sugar, just like S. Then there is P for phosphate group. Then S, then P. There is alternately placed S and P like this. There is one linear chain, and the nitrogen bases are present at the sugar only. Nitrogen bases are present at the sugar only. This I am writing for the DNA, and here at the sugar, let it be the adenine, here be the thymine, here be the cytosine, here be the guanine. Here there can be cytosine, here there can be adenine. So this is the sequence, particular pattern. Now, on the opposite side, there is the same arrangement. So here we have the one purine and one pyrimidine. Here there will be the thymine, adenine, and here there will be the phosphate. So in the same pattern we draw. Like this. Now this is very important part to note, which I am drawing. Now between adenine and thymine, there is always and only double bond. This is the hydrogen bond. This is also hydrogen bond. This is one point. Now other point is between adenine and thymine there is only double bond. Two hydrogen bonds are present. Due to the presence of suitable atoms, that is hydrogen bonding between the hydrogen and other electronegative atom. But between cytosine and guanine, there will be triple bonding, 3 bonds. This is very important point which you have to keep in mind. This is first point. So similarly, like this. Now other point is that the group of phosphate group, pentose sugar and nitrogen base, it forms the nucleotide. It forms the nucleotide, and this sugar and nitrogen base, it forms nucleoside. So this is what? This is polynucleotide. It is called as polynucleotide. Hence we define that biomolecules, that is the nucleic acids, they are polynucleotides. This is one point. Now, very important point, that in any condition, in any nucleic acid, the same base pair is not repeated. Same base pair is never repeated. Same base pair. Like here, it is adenine, it is thymine. This is so here there cannot be adenine. There can be thymine. This is right. Similarly here there can be adenine and thymine. So similarly here there cannot be just here cytosine, here it cannot be cytosine. This is very unique point of the DNA. And you all must knowing the variations. The variations in the animals, organisms are due to this base sequencing. You all must be knowing genetic engineering. So there is a particular sequence of the bases, and this is the reason why the genes till date, the genes or the genome of two persons are not same. This is the reason. The positioning of the base pairs, this is base pairs, is very, there are the millions. In a body, in a cell, there are millions. So there are the millions of possibilities. And as a result, this is the reason behind the variation in the hereditary traits, and no two organisms can have the same base sequence. Even a child is not totally identical with the parents. There are some factors there we can identify easily, but the base sequence cannot be similar or equal, and this is the reason behind the variation. You might know the variations in animals, variations in plants. Variations are produced by this very minute change in the gene sequence results to the abrupt change in properties. That's why the children, they differ from their parents, but they have the different set of patterns. And this makes the unique thing about the DNA and the reason behind this very complex arrangement or the distribution. Now the functions of nucleic acids. So DNA is responsible for the transmission of hereditary characters from parents to offspring. So DNA, it is responsible for the transmission of genetic character from one organism to another. So at the time of cell division, the DNA is copied, and so the child gets majority of the traits of their parents. Then RNA has many functions, but basically the synthesis of proteins. So the protein synthesis in organisms by selecting the required type of amino acid is due to RNA. RNA is responsible for the collection, and it is a very complex process. And so RNA involves different points, and RNA is of different types. These are the classes. So these are the very two important functions. RNA, it is of different types. First is rRNA. R stands for ribosomal. It is responsible for the constitution of the body of the ribosomes. Then mRNA, messenger RNA. It is responsible for the transmission of any information or passing of any information from one place to other place. And then tRNA, it is responsible for the process of the information. So these are the three types of the RNA. So actually neither DNA nor RNA is capable of doing any work, and it is the combined effect of the processes, because however hereditary character is concerned, it is totally the DNA. But this synthesis of proteins, it is made by RNA, but it is somehow helped by the DNA. And so DNA, RNA, they are the fingerprint of any organism, and so they are responsible for the entire character in the trait.